Tack så mycket. Jag kan inte tala svenska längre. Jag ska prata engelska. Okay. So I'm really very honored to be here, and thank you, Jens, for hosting me and all of you. It's really exciting to be back. So why did I come to Sweden 35 years ago? Well, I came to work with this man, Nils Friis. I had studied his work as an undergraduate, and I was fascinated by this concept that plants and microbes could talk to each other, that there was some type of communication between these two very different types of species. And I think Nils was really a pioneer in this area, trying to understand what it was that allowed trees to form mycorrhizal associations. And he hypothesized and did early work showing that there were molecules that were exchanged between the host and the microbe. And we know that this type of dialogue, molecular dialogue, is very common and really very important. So this is arbuscular mycorrhizal associations, and in this case, the fungus actually forms haustoria and inside the roots of the plant, and is able to provide mineral nutrition that the plant needs. Another very famous interaction in agriculture is Mesorhizobium, which is critical for nitrogen fixation. And here you can see nodules formed on the roots of plants. And so I should mention that mycorrhizal fungi associations and bacterial Mesorhizobium associations are beneficial. They're symbiotic. But there are also situations where you have an organism that's a parasite, and one of the really challenging parasites worldwide are these plant parasitic, such as Striga, which you can see here infecting the roots of its host, corn. And these kinds of associations are not limited to plant microbe associations. This is the HIV envelope glycoprotein, which forms very precise interactions with receptors on the human cells. And these interactions are really very specific. Something like 10% of Caucasians are immune to HIV infection, and we now know exactly the molecules involved in that specific recognition. So today I wanted to describe two projects from my lab. The first was the discovery of molecules that control host microbe interactions. So I left Sweden and I went to UC Berkeley to continue this type of work. And then I'll also talk about a project, engineering rice for tolerance of environmental stress. So I work on rice, which is the staple food for half the world's population. And it's very important, especially for smallholder farmers in South and Southeast Asia. So when I began my studies, when I began my work at UC Davis and Cornell, I decided to work on rice because the idea was if you can make just small changes in the resistance to disease or tolerance of environmental stress, you could potentially affect benefit thousands of millions of farmers. So that was really the driving force that drew me to rice. And also I learned in Sweden, it was very difficult to work with pine trees. Okay, so this is a project I've worked on really for quite a long time. What are the genetic components controlling the interaction of the rice plant with a serious bacterial pathogen called Xanthomonas oryzae, Pathovar oryzae. And what you see here is a rice leaf that is infected with this bacteria, and it starts to ooze out of the veins of the plant. It has a yellow pigment. And it's really a serious disease, both in Asia, but also in Africa. and in certain conditions, it can devastate yields up to 50% or sometimes complete crop loss. Now, when I started my work, at the time, it was known that many plants that we eat carry genes for resistance, and it's a very important tool for farmers. If your plant's resistant to disease, you don't have to try to spray something, and you don't have crop loss. So this idea of endogenous genetic resistance has been very, very important in agriculture for over 100 years. Everything we eat probably has been bred for resistance genes. But when I started my work, no one really knew what these genes were. So in the late 1980s and 1990s, there was a lot of excitement in plant pathology to try to isolate these genes. What are these genes for resistance? And I was fortunate to meet Gert of Kusch, who was actually working at CGIAR, which you all know what these centers are now. And he was in the Philippines. He's also UC Davis Emeritus faculty now. And he gave me a package of seeds. So he and his colleagues had been looking around the world for unusual species that had resistance. And he was in Mali, in West Africa, and they found a wild species of rice that had a gene that conferred resistance to all strains of the bacteria that were known. So he gave me a bag of seeds, something maybe 1990, and I decided to try to isolate that gene. So we were successful, and what you see here on the top are a pair of rice leaves that have been inoculated with the bacteria. And it's very simple. We take scissors, we dip them in the bacteria, we clip off the edge of the leaves, we wait 2 weeks, and you could see the conventional variety. You see very long lesions, and that will translate into reduced yield. We isolated the gene through a genetic technique called map-based cloning, and we then introduced it into that same rice variety and then inoculated it. And you could see that that single gene, which we named XA21, conferred very robust resistance to disease. So what does that gene encode? And we were able to predict, just by looking at the predicted open reading frame, that it encoded a receptor kinase. And so for those of you that are not perhaps molecular geneticists, what you're looking at here, and if I could figure out the pointer, I better not push too many buttons, but this yellow line is the membrane. And on the outside of the XA21 protein is a extracellular domain. There's a transmembrane domain and an intercellular kinase enzymatic domain. And so that structure indicated to us that probably that extracellular domain was recognizing something produced from the microbe, and that recognition would trigger a defense response. So that turns out to be true, and I'll explain a little bit of the evidence behind that. But very shortly afterwards, it was a very exciting time because the toll receptor from flies, which had been isolated a number of years ago, was shown in 1996 to also be required for resistance to disease. And the TLR4 receptor was discovered in mice, and that also confers resistance to disease. So you have these very different organisms that have remarkably similar structures. All of them have these predicted extracellular domains in the class of leucine-rich repeats, and they all associate with a kinase domain that are in the same functional class. So at this time, we really learned that plants and animals use very, very similar approaches to recognizing and responding to disease. So the next step for my lab was to try to figure out, okay, well, if this is true, that there's the XA21 protein is binding something from the microbe, well, what is that molecule in the microbe? And we spent quite a number of years trying to isolate that molecule. We also used the molecular genetic approach. So now you're looking at a bacterial chromosome with several genes on this. So there's an operon with the RAXST, RAXA, and RAXB genes. And also, you can see two other genes here, which I'll explain in a minute. But we named these RAX genes because they're required for activation of XA21 immunity. So if we knock out these genes in the microbe and then inoculate the microbe, the plant no longer recognizes the microbe and it's susceptible. It turns out that we've been able, over a number of years, to understand what's going on. So RAXST encodes a tyrosine sulfosyltransferase, and that actually sulfates this small protein called RAXX. And I'll show you how this functions. Inside the microbial cell, we have sulfation of RAXX, and then RAXX is secreted through a type 1 secretion system. So this is this RAXA-B-C type 1 secretion system is a type of system that has been known in many different organisms to take molecules from inside the cell and secrete it outside the cell. So now we had a very interesting situation. We identified the molecule that is secreted outside the cell, and that strains that don't have RAXX are not recognized by the plant, so they actually cause disease. We were really excited about this because it turns out that sulfated molecules are known in many other types of extracellular interactions. So back to nitrogen fixation, in the alfalfa rhizobium interaction, you have to have sulfation of the NOD factor in order for alfalfa to recognize it. If you lose sulfation, you lose this specific recognition. So there's this common theme of recognition specificity that is provided not only by the small molecule, but also by sulfation. Another example is carrot. So this is meristem development. Sulfation is also important for these receptor ligand interactions to have carrot meristems develop normally. And this was actually quite an exciting discovery for my lab, because it turns out that the receptor for the sulfated peptide is also a leucine-rich repeat receptor kinase. And then back to HIV envelope glycoprotein, it turns out that the human co-receptor CCR5 must be sulfated in order to recognize HIV. And you've probably heard of the CRISPR babies, and that was Chinese scientists that were trying to recreate a naturally occurring mutation in the Caucasian population, and it's a precise deletion of the sulfated epitope. And so we know that, at least for the naturally occurring allele, if you are not able to sulfate that co-receptor, you're immune to HIV. So you lose this binding. And I guess it remains to be seen what these new alleles that were introduced through the CRISPR genome editing, what effect they're going to have, because they didn't occur in the human population before. All right, so then the computer's back here. Whoops, uh-oh. Okay, now I'm going to need some help, because there's a lot of buttons here. To the rescue. Thank you very much. I could hit a few, but okay. So then our next step was to figure out, does this secreted peptide actually interact with the receptor? And we were fortunate to work with Yosef Belkadir's group at the Gregor Mendel Institute. Thank you. And we were able to show that this sulfated RAXX does interact directly with the receptor. So what you see here is Xanthomonas, and you see RAXX is secreting the sulfated molecule, and that binds to the receptor. And we have a number of markers for resistance. Of course, the plants are resistant, but also we can measure other types of biochemical events that allows us to more quickly assay these interactions. Okay, so why does a pathogen produce a molecule that allows it to be recognized? So if you're an evolutionary biologist, you would say, well, that really doesn't make sense. And so for a long time in the field, the idea is, well, the pathogen must be producing this molecule for another reason, somehow to enhance the fitness, its own fitness. And we still don't have the answer to that, but we did observe that the RAXX protein, the amino acid sequence, is very similar to endogenous peptide hormones in the plant. So almost every plant species carries a peptide hormone called PSY. And we have the Arabidopsis PSY, Oryza sativa, rice PSY, and then the RAXX molecule from the microbe. And you could see they are very, very similar. And we know that the plant PSY is also sulfated, and it's also sulfated by tyrosine protein sulfosyltransferase. So what is the function? That's where my lab is now. We've been able to show that when the microbe is lacking this RAXX, it's compromised in its ability to infect the plant. So we do have evidence that RAXX is important for the life cycle of the microbe. And so what we hypothesized then is that this microbial protein facilitates bacterial infection. And somehow the microbe is targeting what we call the PSY receptor, which has not yet been discovered, but we hypothesize it's also a receptor kinase, and that is the same receptor that the native PSY is targeting, and that's leading to growth and development, and somehow that's facilitating infection. But plants are not stupid, and over the years, rice has evolved the ability to recognize the microbial version. And that recognition leads to an immune response and stops infection. So we are now carrying out experiments to try to isolate the PSY receptor and to understand mechanistically how that RAXX protein is facilitating infection. So that's the first story. So immune receptors in plants and animals are very, very similar. And also we know that the bacterial pathogen secretes a peptide hormone, and we believe it confers a selective advantage. Now, that's work really building on work I started in Uppsala, but I am interested in rice more broadly. And as you heard from previous speakers, we have a major issue with climate change. And these are hydrological issues. So not only do we have heat and drought in some regions of the world, but we have flooding going to have increased flooding in other parts of the world. And the Intergovernmental Panel on Climate Change predicts that flooding is going to increase in severity and duration in many parts of the world, including Bangladesh. So what you're looking at here are 4 children walking through their family's rice paddy in Bangladesh after a flood. Now, the rice on the outside will continue to grow because most rice varieties grow well in standing water, but most rice varieties will die if they're completely submerged for more than 3 days. So in the center of the field, the farmer is not going to be able to harvest grain. And this is a major problem in parts of Asia. It's estimated that 4 million tons of rice, enough to feed 30 million people, is lost every year to flooding. So my colleague Dave McKill at UC Davis approached me after we had isolated the XA21 gene to see if we could use the same techniques to isolate a gene that he had roughly mapped for submergence tolerance. Okay, so this shows you where 25% of the world's rice is grown, and there are 70 million farmers in this region that live on less than $3 a day. So it's been a big challenge. It's been something that breeders and farmers in this region have been working on for a very long time. And these international centers are really very, very important because not only do they do great work, as we heard about in the earlier talk, but they also store genetic diversity in the form of seed. So the International Rice Research Institute, they have over 80,000 different rice varieties and rice species with lots of different traits. Most of these are not even grown anymore. And so scientists at the International Rice Research Institute screened through part of the collection, and they found an ancient rice variety that was highly tolerant of submergence, and it originated from eastern India. And breeders had tried for a number of years to use conventional breeding to introduce this trait into varieties grown by farmers, but the farmers rejected those varieties because when you use conventional breeding, you mix lots of genes together. It's not very precise, so the farmers then are losing other traits that they're very interested in. So Dave and Kenong Xu, his student, and I decided to try to isolate this gene using a bat-based cloning approach. And we generated very large populations segregating for this sub-1, submergence tolerance 1 region, and identified markers. And then my lab created very large bacterial artificial chromosome libraries. And so you're kind of now creating pieces of cloned DNA that are spanning that region. And then we sequence that region. So this is a typical mat-based cloning approach, and so for some of you younger scientists, this was the day before we had any genomic sequences, so we had to do this, sort of by hand. And we were fortunate to identify in that region a group of predicted transcription factors. There were 3 of them, sub1A, sub1B, and sub1C. And we immediately started looking at those because we knew that they had been previously identified as to be important for stress tolerance. And also, we found very quickly that the sub1A gene was rapidly upregulated upon submergence. So that gave us a hint that sub1A was the key gene. So we took that gene and we introduced it into rice varieties using the same technique of genetic engineering. And what you see now on the left is a conventional variety of rice that was grown for a couple weeks, and then it was completely submerged for 2 weeks, and then it was allowed to try to recover. But it did not recover. And on the right, you see the variety that we developed that carries the sub1A gene, and it's very robust. So this was very exciting to us. It indicated that we had probably the key gene that conferred submergence tolerance, but this was just a laboratory experiment that we carried out in my lab in greenhouses. And really the question is, can this be used to benefit farmers? So Dave McKill had left UC Davis, had gone back to the Philippines, and carried out a very important long-term breeding program where he used another genetic technique called marker-assisted breeding to introduce this gene into many varieties grown throughout Asia. And I think, okay, I want to just say that before I get into the applications, I want to say that we have had a wonderful experience with this project, because not only did we have this laboratory result showing me they had the gene, but we have a colleague, Julie Bailey Sears, and a number of other colleagues that really dug down deep to understand what the mechanism was. So how does a single gene confer submergence tolerance? So it had been known for a long time that when rice plants are submerged, it creates a low oxygen and sort of a local accumulation of ethylene. And what Julia's group and also several other people shown on this slide were able to show that local accumulation of ethylene upregulates another member of the gene family called sub1C, and it leads to cell elongation and carbohydrate breakdown. So what this means is that in normal situations, rice plants will try to grow out of the water. And often they will die because they deplete their energy reserves. But in the presence of sub-1A, so you just need to add a single gene, what happens then is sub-1A binds to a set of transcription factors that are stabilized under low oxygen, and that leads to activation of ethanolic fermentation, which provides an alternative energy source. And sub-1A also downregulates the sub-1C pathway. So now the plants are not trying to grow out of the water, and you end up with submergence tolerance. So the sub-1 locus is really activating this survival pathway during flooding stress. Okay, 2 minutes. So I just want to finish by giving you the practical application. So this is David McKill's work in the Philippines, and what you're looking at now are rice plants growing in the field in the Philippines. And on the left, you have the sub-1 variety. On the right, you have the conventional variety. They both grow well at first, but after a flood is applied, you see that the sub-1 variety grows very, very well, whereas the conventional variety is dying. And I like to show this slide because, as most of you probably know, there's a lot of anxiety about genetics and questions about why are scientists doing this, and I think it really gives a graphic representation of the benefit to farmers. And this has been the Bill and Melinda Gates Foundation have helped distribute and bulk up the seed. Dave has led a number of field trials, and we see a 60% yield advantage in farmers' field. So you can see Swarna sub-1 is shown there on the right. And then there's economists that have been looking at us, well, who benefits? And what you're looking at is a region in eastern India, and the light yellow there, the light pink, are low-lying areas. And what Kyle Emmerich has found is that these low-lying areas tend to be more heavily occupied by lower caste social groups. So the generations of very, very poor farmers have had their lands flooded, and they are disproportionately benefiting from this rice and are able to grow rice even in some cases where they would have lost their whole crop. So last year, 6 million farmers grew sub-1 rice. And that's really what I wanted to tell you today. And just to remind you, plant genetics is an important component of sustainable ag. I think often in the public dialogue, there's this really distraction about genes in our food. And I really believe we need to get back to the critical aspects of sustainable agriculture and ask how are we going to help enhance food security and nutrition for the poorest people in the world, minimize environmental degradation, reduce toxic inputs, keep food affordable, and foster soil fertility and biodiversity. And thank you to Nils and his family. It's been 35 years, but I think I recognize Eric in the audience, who was there with the big grin on his face with his daughter Nina, and that's Nils' wife Elizabeth, and they were really wonderful to me, took me on mushroom hunting expeditions, and really had a high level of how to do science with integrity and good humor. So thank you very much.